CCMB. I have great pleasure today of introducing Jake Scott, whom I first met through Brandon Ogbunu, who was a former colleague of ours here. And the context there was a really neat paper that was on its way to coming out in Nature Physics, using theory from physics, an apparatus called contradiabatic driving, to try to imagine controlling the distribution of evolutionary trajectories that a population is going to take from one genotype to another as it responds to the contours of the fitness landscape. So this is a stochastic process, and if you imagine 1 million replicates, there'll be some distribution of paths that are followed. And Jake and his collaborators were interested in understanding whether they could manage or influence that distribution by changing the environment in a thoughtful way, changing the environment in time in a thoughtful way. It was really cross-cutting. Jake studied at the Naval Academy, studied physics at the Naval Academy. He served on submarines before going to medical school and becoming an oncologist. He has a DPhil from Oxford, bringing ideas of population genetics and spatial heterogeneity to clinical problems like cancer. The work that I just described was in antibiotic resistance evolution, and he's really, to me, kind of an amazing engine of innovation, and also sees patients. So without any further ado, thanks for making the trip. Oh yeah, sorry, let me acknowledge Jake Margulis, who was an undergrad in my lab and is now in the MD PhD program here doing his PhD with Jeff. And it was totally Jake's idea that we should invite Jake. So credit where credit is due. Yeah, take it away. Thanks.
Or blame Jake if you don't like the talk. Thanks so much. It's a real honor to be here and meet everybody and be a part of this group. I have also had a fantastic day, so thanks everyone for hosting, and the student group that led our journal club, which was a really cool idea. I'm going to steal that. Okay, so I'm going to spend close to the next hour talking about. We're going to have a talk in 4 parts. First, I'm going to open up with a little bit of my philosophy about evolution and ecology and why we should care about it in cancer. And I'm also going to say an infectious disease. I'm an oncologist, but I think all the things apply here to any evolving disease or population, really. And we're going to go back and forth between experiments in bugs and experiments in cancer. We're going to talk about some mathematics. We're going to talk about some simulations. We're going to talk about some sequencing. We're going to talk about some stochastics. So we're going to go all over the place. I'm also going to talk a lot about timescales, because I think it's really important that we focus on those. Specifically, what's fast and what's slow. And I'm just reminding, I'm also going to go through parts of the talk more quickly and more slowly, because there's some of the details that don't matter, but I want to get the ideas across. And I was reminded by my son that if I was playing a musical piece, I would be using the term called rubato. So I'm going to slow down and speed up as I go through the talk. There's lots of details that I'm going to gloss over. I don't think they matter for the ideas. If you want to talk more about them, I'd love to. But I'm going to try to cover maybe a little more ground than I should have, but I'm really excited to be here with this group and want to share a lot of some of the work we've been doing. I'm also going to spend just a moment introducing my group and our group's mission, because I think it bears repeating and reading out loud in these times. So this is my group. I'm really happy. This is at a moment in time over the summer. And I wanted to read the statement that we have on the front of our website. I'm going to read out loud. Our lab's committed to creating and maintaining an environment in which all are welcome and respected, one that's inclusive of race, gender, faith, sexual orientation, ability, and socioeconomic status. We acknowledge many identities are underrepresented in STEM and in academia, and that systemic racism and other discriminations create obstacles for students and postdocs. We strongly condemn behaviors and institutions that perpetuate these inequities, and we as a lab and a community must act to dismantle the structural and systemic barriers to higher education. And further is our mission statement there, and I want everyone to know that regardless of what's going on in the rest of the world, I'm not changing track, and I hope we don't either. Because I think we're stronger when we're together, no matter what. And diversity in opinions and faces and backgrounds matter. And there's a lot of homogeneity in a lot of our fields, and it's because of systemic issues that have perpetrated this for a long time, not because of the hearts and compassionate minds we have in this room. Anyway, just want to start there. Okay, so we're going to set the stage. I'm an oncologist. With the exception of that little blip we had a few years ago, cancer is the #1 cause of death in the developed world. In the world where we're still developing further, we still have a huge problem with infectious disease. And obviously we did on the whole globe just recently. So the World Health Organization, when we were a member of that, listed these as the 2 top parts of our problem in this era. But I think, I don't want to doom and gloom the whole thing. Most of the time infections are cured with antibiotics or just waiting and getting some sleep because our immune systems are amazing. And further, the vast majority of cancers are cured, early stage cancers. You know what the #1 cancer cure is? It's a knife, the inclined plane. Surgeons cure more cancers than anything. And so even these simple ideas still suffuse. That said, once infections become, this is just a picture for Jacob. This was a patient we talked about a little earlier. This is actually a 22-year-old man who I treated with a tumor. This is his heart, and this is a tumor right on top of his heart. And we were able to, in 5 days, with no side effects, get rid of that, and he lived another couple years. So we do amazing things. However, once these diseases spread distantly, we fail always. And so this is a picture of a gentleman who I would say is we considered lucky because his melanoma, which had spread all through his body, had a specific targetable mutation. And it doesn't matter what it is, but it's called BRAF V600E. But the good news is that we have a drug that targets that. And after 4 weeks or 15 weeks, 4 months of being on this drug, his tumors have gone away. However, evolution finds a way, as Jeff Goldblum told us in Jurassic Park. And this failure of targeted therapies is conserved. It's not just here in personalized oncology. This is the same problem that causes us to have problems with monoculture in farms, where we use the same insecticide every year, problems with antibiotics of various kinds, invasive pests, viruses. So I would say every kingdom of life experiences this issue. Monocultures fall down. And we struggle. And in cancer in particular, there's lots of reasons why this doesn't work. But a meaningful paper to me from a mentor named Bob Gatenby really put out this idea that evolutionary dynamics sort of show us right off from first principles that this idea of personalized medicine should fall down. That said, the entire ship changed when we invented Gleevec or imatinib. We talked about this earlier. I think it's actually a little bit of an accident of history. The first targeted therapy we ever invented was for a disease called chronic myelogenous leukemia, which is a blood disease. And there's a pill you can take called Gleevec, which targets a specific protein. And this is a disease that evolves slowly. So we invented this medicine, started giving it to people. It was targeted to that one thing, no side effects, hardly. Diseases went away, but it's a slowly evolving disease. So it took us about 10 years to start seeing evolution beat it. In those 10 years, the entirety of oncology changed the direction of the ship and started focusing on personalized medicine as the cure because we thought we'd done it. All we have to do is find the one aberration, design the one drug, and off we go. I wonder what would happen if the first targeted therapy we found was lapatinib or one of these other ones that fails quickly. Would we have blinked? Would we have even turned around? Would we even have personalized medicine? Would we be better off if we didn't? I don't know. The entire engine and economic engine and academic engine changed to this, I think, doomed to failure model. And I think you can do this from first principles. Here's a paper from a friend of mine, Mike Boehm. Many of you have probably seen this. This is the mega plate experiment. If you haven't watched the YouTube video, do it. What's happening here is a 10-foot long plate. This is actually a huge petri dish. Was inoculated with drug at, well, no drug, and then 3 times the minimum inhibitory concentration of the bug, 30, 300, and 3,000 times. And this is just a simulation a student of mine, Nikhil, did of the same idea. You basically put E. coli here and then wait a week. You wait a week, which, by the way, E. coli to go 4 feet is like us going to the moon and back, right? So for these guys, not only are they fast, but they evolve resistance to 3,000 times the level of drug that should have killed them in a week. And they didn't do it once. They did it thousands of times. Amazing numbers of times quickly. So to me, this experiment by itself tells us that personalized therapy is never going to work, right? I give my cancer patient a drug. It's going to figure out thousands of ways to beat it. And I'm never going to catch up. I'm going to end up like Alice when she's through the looking glass playing croquet with the Red Queen, staying in the same place, just as running as fast as she can. So we're in this trap, I think, where we're constantly chasing this algorithm that's beating us. And here in this experiment, mutation and evolution are fast processes, right? This is what's driving, there are certainly some clonal interferences physically here, but for the most part, this is an evolutionary process I think is happening over the course of a week or so. I'm going to contrast that with a different idea. And I want to talk about a cognitive dissonance that I experience as a physician scientist. So when I come into a room with a patient, they're sitting there, they have a tumor, it's causing them pain, it's threatening their life. They don't really care if it's evolving or how it started or what this beautiful evolutionary process that led them to today was. They just want to know what to do, how to make it stop hurting. As a scientist, I'm fascinated by the months and years that led up to this, right? So this is a diagram I'm going to show in the next few slides. So I want to tell you what this picture means. This is a concatenation of a Mueller plot, which many of you may have seen, which is a plot that describes population size and clonal content. So each color is a clone or maybe a species, if you'll allow me. And the height of it is how big. So this is a bigger tumor than that. And this is a more heterogeneous tumor than here. Patients only care what to do now, right? The minute I put a needle in it and ask what to do. But over the course of this time, a lot's going on. And I'm going to talk about each of these timescales in particular as we enter the talk and talk about them. But I want to focus for a moment here on what I consider to be like a clinical timescale. The weeks and months coming up to and right after this. But we already have an established heterogeneity. There's an established ecosystem. And I can perturb it with drugs. Now, let me ask you a question. If I come to a game reserve and I have some number of wolves and moose, and let's say by accident of mankind coming in and digging holes and screwing up the environment, the moose population has been crashing. What's going to happen to the wolf population? It's going to go down too, because they don't have enough food. But at the same time, there's a possibility you have an extinction event, right? If you have an extinct wolf species and there's no more food for the wolves, what happens to the wolves? They're also going to crash, right? But if I can go in and bolster that moose population somehow, bring some in from the outside maybe, or stop some sort of land destruction, I could stabilize those populations. I could bring them back up. Now, do I have to have the moose evolve for that to happen? Do I need a new mutation to come into the wolf or moose population for the population dynamics to change? No. As a matter of fact, it wouldn't matter, right? I bring in a really super healthy wolf today, maybe some crossbreed that's faster or stronger. Is that going to massively affect the populations next season? No. The next season? Probably not. The next season? Probably not. 10 years from now? Maybe. But what I'm getting is that the ecology here is fast and the evolution or mutation is slow. And when I'm saying fast and slow, I'm being a little bit fast and loose with terms, but I'm saying the more important term in this setting is the ecology. The less important is the mutation. It's very different than the one I just showed you, right? Where the mutation was driving everything and the ecology wasn't. What's fascinating to me, and this is obvious, right? So we've got bugs, we've got wolves. Cancer's kind of in the middle. Both are at play. And so we have this situation where we have a pathologic, natural, but not real life form, where the admixture, sorry, that's the wrong word, where the timescales of ecology and evolution are truly starting to overlap. On timescales where I can perturb either, and my knowledge of each can lead in. But we often think of them separately. Okay, so that is leading us to this big plot that just shows that I think cancers and infections are both complex and adaptive systems, and they change and adapt over multiple timescales. We're going to focus on two of those today. I'm just showing you this picture because our lab thinks about a lot more. But we're going to talk about the first one there, biasing selection, and the second one about harnessing ecology. Okay, that's the end of the very beginning. I'm going to go through now the timescales and just how I think of them, and then we're going to dive into some science. I'm going to pause in a minute and ask if there's any questions about the philosophy, and then just barrel ahead. Okay, so I'm going to describe each of those timescales a little bit more in detail. So this, and I'm going to name them, not perfect naming conventions, but it's going to be a naming convention nonetheless. So imagine this evolutionary timescale, which is sort of the slowest, if you will, which is what's driven by evolution. And and I say evolution, I mean mutation. So what happens when populations fundamentally change, when we have new speciation events, when we have changes in that population structure based on literal new events. So we often think about this in terms of evolutionary branching points or fitness landscapes. People use adaptive dynamics. There's a lot of ways you can model this. We'll talk about some. And then here are some of the folks in my lab that I've worked on with this, and you'll see some of their work coming up soon. And here's actually that paper that Dan was just chatting about. So it's led us thinking about these concepts. The middle time scale, which I'm going to call the clinical time scale, where I think ecology dominates, is listed here. And we're going to talk a lot about a tool called evolutionary game theory. This is a student of mine called Artem, who's now in Utrecht. And on the upper left, we'll come back to Jeff in a minute. He's on the job market, but he's fantastic. And then there's this now time scale, which we're not going to talk about today, but I think a lot about. As a matter of fact, I'll be giving a lecture on this at the Cancer Center tomorrow. So if you're interested in learning more about how I think about basically GP maps of therapy sensitivity or polygenic risk scores or polygenic response scores to radiation and chemotherapy, I'll spend the whole hour tomorrow talking about a series of papers we think about there. Very, very different topic, but still probably part of the same family of thoughts. So personalized chemo and radiation dosing. Not today. Okay. That's the end of the beginning. Any questions about anything so far? Probably not. It's just some philosophy. All right, rock on. So our group, we call ourselves Theory Division. It's a joke about a band called Joy Division. Does anyone know the album cover? It's actually Pulsar data for the nerds out there. And I've got a great story I can tell anyone afterwards about a personal hero I met over that t-shirt. And in our group, so I'm a physicist who did applied math and medicine and engineering, which means I'm nothing, which means our group uses any tool that's appropriate. And I think that's important here because there's really, really hard problems and we need to have a diversity of tools and questions. So we use things called cellular automata, which we talked about today in the student lecture. We use systems biology and informatics if we need them because they analyze data as we get it in these evolving populations. We're going to use game theory, which we're going to talk about today. We think about stochastics, landscapes, and seascapes, which we'll talk about today. We grow stuff in the lab. We stick needles in patients. We do all kinds of stuff. It's a mess. It's a beautiful mess. Okay. We're going to dive into this one time scale. The next 15 or 20 minutes are going to be about how to think about ecology and cancer, and in particular, this clinical time scale, and ask the question, how does interaction between members of an existing standing heterogeneous population affect fitness, individual fitnesses, and drug response? And again, we're going to talk mostly about a paper here that we wrote a few years ago, but there's been a bunch of follow-ons since, which you can see in the upper left corner. Okay, off we go. Yeah, cool. Okay, so we're going to frame the question, and I think this group probably doesn't need as much of an introduction to this as many. When I first started my oncology training, most oncologists thought of cancer as a single ball of mutated cells that were all the same, that were growing wrong, that had the same founder mutation, and off they went. We've since come to realize in the last 15 years that that's silly. This is actually this beautiful, I would say, heterogeneous ecosystem that has normal cells, blood vessels, fibroblasts, stroma, cancer cells of all different kinds, immune cells coming in, all making up what is now we call a tumor. This is very similar to a complex ecosystem. Theoretical ecologists have been messing with hard problems with multiple species together for a long time. Food webs, things like this. There's an established mathematics of this. I think something that's really interesting, and your friend this morning, Chibu, I was talking to, came up on this idea, really is, there's a huge complexity here. And if I sequence that, if I stick a needle into that, and I pull out a genomic sequence, I'm going to get information about lots of stuff, not just cancer. How much detail do I need to know? And we're going to talk a lot about that. If I go to the national forest, and I measure the wolf and moose population, I'm ignoring the fact that there's 15 different grass species that the moose also eat. And I'm ignoring that there's rodents that the wolves also eat. I'm ignoring that there's insect species that prey on the rodents by drinking their blood. Do I need all that? It's there, it's real, it's part of the ecosystem, but do I need that level of information? Or is it good enough just to know that there's wolves and moose? In that situation, I think it's probably good enough to know there's just wolves and moose. What kind of what level of resolution of control do I want and need? What level of resolution of measurement do I want and need? I think in biology, we're often stuck because when I stick a needle in that tumor and I get a sequencing back, I get 23,000 genes. Do I need all those? Maybe it's just clouding. Maybe the signal noise problem is really there, which is where I think theory can help us. Because if we come in with a parsimonious theory and then ask what that tells us to look for, we now have a guide to deal with that data. So what we're going to do is we're going to go back to some of these old ideas in theoretical ecology and see if we can reproduce those in cancer in some way. So these are some beautiful papers, again, by a mentor of mine, Bob Gatenby, really looking at how we can apply, those are some Locke-Volterra-like equations, really just competition, predator-prey dynamics, to really model some of these situations quite beautifully. And I'm going to show you a couple slides of where we've done similar ideas, but with cancer, just to see what kind of things we can come up with. So this is one of my favorite papers because it was my gateway drug. This is the first math paper I was part of, and it sucked me into the world, and I went on a 7-year journey that my wife does not rue, but it took 7 years off my life. No, it added 7 years of joy to my life and brought me to where I am in this world. And so what we did here is we took a game theoretic model. So this is a discrete time replicator equation. Details don't matter for this slide, so don't get bogged down in the equations. The idea is we have some sort of iterative process that goes forward in time, and we have some sort of payoff table that tells us about how to calculate fitness. From this, we can look at how these populations, blue, orange, and green, don't worry about what they are. We can look about how they change over time. So this is deterministic. Nothing is stochastic or crazy here. It's all once you write down the model, you get the answers. But what we found is when we intervened with a therapy, could change the outcomes massively. So an early intervention makes green win. A later, same intervention makes blue win. A short time intervention, early, green still wins. A longer time intervention, blue now wins. So we can sort of start changing the outcomes of these. But to a mathematician, well, these are great. The questions we can answer are when and how long. But to a mathematician, it's sort of obvious because you wrote everything down at the beginning. It's a deterministic system. If I tweak it early, I tweak it late. No big deal. This is just simply a dynamical system with a couple different attractor states. You're moving between one basis of attraction or another. Mathematicians are like, so what? However, you can do the same thing with different ideas for sequencing. You write down this model. You can ask the question, what order do we treat? So bringing these tools to bear on a cancer system, again, is not mathematically new, but it's biologically non-trivial because we can start applying tools we didn't have before. But again, this depends on what you wrote down, your assumptions about the system. Once you write down your assumptions, the rest falls out, right? There's no surprises here mathematically once you've written this down. So the most important question is, wait, does any of it matter, right? So I've shown you I can do stuff mathematically, but does it really matter? And the answer, I think, is now quite strongly a yes. So clinical trials have been written recently by the Moffitt Group that have shown that clinical trials based on these ideas of ecological control can give us positive outcomes. So in this situation, this is a clinical trial of men with prostate cancer that had spread to their bones, who were given a specific medicine. And the question here, or the thesis here, was that the medicine was killing the sensitive cells, but a resistant population were rising. And if you treated for too long, and one more thing that's important is that the sensitive cells would actually keep down the resistant ones by competition. There was a certain amount of space and nutrients. Sensitive cells would eat it all up. If there was a lot of them, the resistant would stay down, but as soon as you remove the sensitive ones, the resistant could rise. It's an idea called competitive release in ecology. And the idea here would be instead of giving that medicine entirely forever, you give it a little and then stop. Let the sensitive rise back up and let them control the resistant cells themselves. There's a whole bunch of wonderful assumptions cooked into this model. It's not my own. You can read my thoughts on it as a reviewer number 2, who's anonymous, but they were published. I have some fundamental issues, but I think in general, what we learned from this is that there's a whole lot of potential. But again, it boils down to what they wrote down in their initial assumptions about that model. So I came to all this and I thought to myself, gosh, it would be great if we could measure that payoff matrix. Because if we could just parameterize that, now we have open to us all the tools of theoretical ecology to start using in cancer. So we asked the question, can we design an assay to directly measure that game being played? And so these are my collaborators. That's Andrei Marusek, who's a fantastic scientist at Moffitt, David, biologist at Moffitt, David Basanta, who's a game theorist also at Moffitt. Here's Artem, my student, who's now in Utrecht, and that's Jeff Peacock, who's now a radiation oncologist in Alabama. And what we did was we did the simplest thing you could imagine. We took the one leading term, resistant versus sensitive, ignored everything else. So we got a bunch of cells that you can buy from the store, the green ones. that are sensitive. They have a specific mutation. They're a lung cancer. They have a mutation that we have a drug for. Don't worry about the rest. We put them in a dish. We gave them that drug. We waited 6 months. Guess what? Darwin was right. They become resistant. And we use the magic of biochemistry to stain these ones red and those ones green. And now we have an ancestral population and a resistant population that are darn similar, obviously different, but darn similar. And then the importance is that their phenotype is different between sensitive and resistant. And they're red and green, so we can count them. And now I'm going to show a little math just to give you a sense of how we're thinking about this and why. All those plots I showed you in the game theory are just ODEs. And they can be thought of very simply in terms of this payoff matrix over here. And so what you can do is you can write down an ODE for the parental, and it only has 2 parameters you need to worry about. So that ODE looks like this. The number of parental is simply this ODE with A and B. These are just numbers of each, and the same for the resistant type. And you can simplify that down because we make some assumptions about constant population size, which is a bad assumption, but we'll deal with that later. That's how I shoot. And so what we're stuck with is a problem of how to get 4 numbers from some system. And so what we started to do was ask the question, what if we co-cultured these cells? So this is the obvious, easy co-culture experiment. We give resistant and green resistant cells and parental cells together. We give the drug. You wait long enough, resistant wins out. That's not good enough. That's the what goes up must come down version of Newtonian mechanics. Not wrong, but not super helpful. So instead, what we did was we started counting and quantifying. So here's an example of an image from a microscope where you have green cells and red cells. And what we did was we set up an algorithm that counted every 20 minutes. So we took a picture, counted the number of cells over the course of a number of days. And I'm going to build a complex plot for you to show you where these parameters come from. And you're going to see a couple things. There's going to be yes drug. So here's a lot of it. And no fibroblasts. So fibroblasts are a normal tissue that inhabits tumors. They can help things or hurt things. Consider it like a season. This is like winter with drugs on. We're also going to look at summer with drugs on, winter with drugs off, winter with all 4 combinations. So what you see here is a count of number of cells, and then later on a count, and then everything in between. And we have this very beautiful, surprising linear fit. So what we really see, and this is a semi-log plot, so it's actually exponential. But what we see is that a linear fit works really well. And so now for the drug with no fibroblast situation, we have growth rates. So we can derive a rate for an exponential growth here. We can now do this for all 4 of those scenarios, with and without drug, whatever system. So here's a different example of this is now with fibroblasts, without drug. So sort of the opposite example. And now it's going to get a little busy, but bear with me. What we can do now is look at 4 situations. So on the far left is drug with no fibroblasts, then drug with them, then nothing at all. So this is a vehicle control for the biologists in the room. And then this is a vehicle control plus the fibroblasts, so plus a normal tissue. So this situation here, the purple one, is like your untreated patient. There's healthy tissue around and there's no drugs. And as we go to the left, we sort of start looking at those different scenarios. And the way you look at this is that the major axis, the major y-axis is the resistant growth rate. The major x-axis is the parental growth rate. And what you can see actually here is the 0, 0 is here. So everything up and to the left is the resistant growing really well because they're above 0, and the parental dying. Makes sense. That's what our drug is on. Everything over here is both growing. So in the absence of drug, both populations grow. And in the absence of drug with healthy tissue, the sensitive are actually winning out, which is a lot like an untreated patient, right? We have a scenario where there's a tumor that's growing really well. Off we go. And now pulling this, so I'm just going to take this single convex hull, which is just the drug situation, and pull out those data specifically and show you where our parameters actually come from. And so from that, that's the same picture. I didn't go too fast. This picture on the left is just going to move up. So it's all these data spread out across this initial proportion cell. And what we can now do is go back to our initial set of equations and start pulling off these intercepts. So here I have all P. And so in this situation, now this situation, all P is none of the R. So this goes to 0. Now I can pull out that A term and I can do the rest for each of the other intercepts. And so what we've done with this experiment, because we've cultured them in different proportions, so that's where these things pop out, is measured the first payoff matrix for an evolutionary game in cancer. So all of a sudden, we now have the ability to use all those tools I showed you earlier, those dynamic systems, tools from dynamical systems, to start playing with cancer models, which I think is exciting. If you now put these into a game space, you can see how these different scenarios play out. So down here is what we do to patients. We give them all the drug all the time. And what happens? Parental goes to all resistant, the tumors become resistant, and the drugs stop working. Up here in the scenario, we have a coexistence, and we have the only situation that's coexistent is our untreated patient, which makes a whole bunch of sense. And so what we see is that adding our normal tissues, we know this from many other experiments, normal tissue actually protects you a little bit from drug.
If no drug at all, we will still have persistence of resistant.
Here, yeah, this is darn close to sort of a neutral case.
What drug?
So that's a super nuanced biological question. In this case, we think it's a secreted growth factor that sort of abrogates the drug response in some way. We've dove into that in other works. It's
not just
no, that also could be part of it for sure. But there is a protective effect that's well known with calves that I'm not exactly. It might be actually different for each evolution experiment.
Because if you do like that you just shift everything down, so
Resistant doesn't mind it too much, but the parentals shift towards resistant more. So their death rates go down. I have a slide I can show you later.
Okay.
And so the question I'm going to ask from this is that, can we think about drug as an evolutionary perturbation rather than a way to kill stuff? Can we think about it as I use the word evolutionary, ecological perturbation. So can we think about it as a way to intervene? It's not just, it is changing some of the fitnesses, but it's also changing that frequency dependent fitness effect. If we measure this in different drugs, we see these evolutionary games popping up all through these spaces, which is wild because once you open this Pandora's box, the evolutionary dynamics in each of these places is wildly different, which begs the question, can we think of drugs in this new way to steer evolution? Can I stop thinking about a drug that's just going to kill it? What's my EC50? What's my IC50? Which we do all the time. And instead say, hey, I understand the dynamical system that's driving this tumor. Can I start playing with it? Can I give things in different orders to play with those systems?
Can you just walk us through a little bit?
Yeah. Okay, so this is that same plot. So this is so each quadrant's quite different. So this is an unstable, this is a bistable system. We've actually never observed anything here. I'm not sure it's observable, but that's a mathematical question we talk about. I'm excited about that. Up here, this is all, it goes to all ancestors. So this is pushing back toward all sensitive. This is pushing toward all resistant. This is coexistence. And we see that depending on the dose of a drug, you're actually moving through this space in different ways. So in this drug, it happens to be cisplatin, standard chemotherapeutic. We're pushing it toward the ancestor winning. So it actually beats the resistant ones better. Whereas lapatinib, which is a drug I was making fun of earlier, pushes things down and through kind of in the way we want to go between all ancestor and all resistant. And what's the other one? Osimertinib does the same thing. So this is actually the first line therapy for this kind of cancer right now. And the cancer biologists in the room might think I'm cheating on you here because this is actually a different set of cell lines that are EGFR positive rather than ALK positive, but same exact setup. I took the cells, I evolved resistance to do the same exact thing. I just didn't mention that.
So this cisplatin is never like the resistant cells are getting killed.
Yeah.
Are being outcompeted by the sensitive cells in spite of the drug being.
Yeah. And we don't give that drug in this cancer, which we empirically came to, but
Right.
it's a fourth line thing that you might give. But again, the evolutionary dynamics change in these ways. And I'm wondering, should we reframe our thinking to be an ecological one? Please.
So the idea that those two drugs that push basically push the balance into the bottom right quadrants, stuff that we want, and then that which stays in the top left quadrants that we don't want to use to treat.
The idea would be, in a perfect world, you alternate between drugs down here and drugs up here. And so what you do is you push it toward a sensitive state, but remember, it's still growing. So you have a tumor that's growing, but it's sensitive. So then you give a drug that pushes it this way, but it shrinks the tumor like crazy, but the resistance starts to grow out. So you kind of have this population where modulating it, where these guys, the sensitive guys, are modulating these ones, and your drug is modulating these ones. So you beat it down, but not so far that you lose control. So you're constantly playing with those populations.
So the idea kind of is you're oscillating, like you're pushing it down, like you're trying to kill off a lot of the sensitive cells, but then you want to use some of the other drugs to go and push it back into that
Yeah.
state where you then back down.
Exactly. And in a perfect world, it becomes sort of a more chronic situation.
But Bob would just remove the drug.
He would take it off. So his pushing up here would be no drug.
Yeah.
Right. And to be fair, in that trial, there was Lupron all the time, and it was on and off of a different drug. So it was on both on one, on both on one. Cool. Okay, that's the end of the first chapter. So we're going to look quick summary. I think evolution drives drug resistance partially through ecological interactions within tumors. And I think that we now are armed with an in vitro assay. There's a whole bunch of beautiful stuff we can do. We have the mathematical tools already. We can start measuring these ecologies through time. Importantly, there's a new journal that I want everyone to know about called PRX Life. It's an APS journal, so it's for the American Physical Society. So it's open source and it's nonprofit. And it's shooting for impact factors like PRX, so 10 to 20. We actually got their first ever cover for this guy, Jeff, who's on the market. So keep your eyes peeled. He's a very talented theoretician and experimentalist biophysicist. And he is shortlisted at Stony and University of Maryland. But if you know someone looking for someone, he's on the market and he's great. And he drove this paper and the one that I just showed, all those things going around and around and around. Okay. That's the end of the beginning. So, okay, I'm going to switch timescales now. Any lingering questions about the ecological one? We'll come back full circle in a minute, please.
What time frame?
Yeah, the middle. So what I would call the fast ecological timescale. But remember, I'm also making the argument that in cancer they're squished together. So this is now going to be the mutational timescale. So if you're going to think about writing down a master equation for this in wolves and moose, this one would be an epsilon that approaches 0. Whereas in cancer, I think the epsilons are approaching each other. Okay.
Earlier, cancer, in that time frame, both the time scale of ecology and time scale of mutation are both somewhere in the middle between fast and slow.
Yeah, in cancer, that's right. They're coming together, and different cancers probably act differently, but in general, that's why it's so fun, because that's why it's also so complicated. Cool. Okay. Dan, don't get too excited. This is gonna be the part you're gonna get some Alan Alda vibes on. I want to tell a little story. So does anyone have one of those moments like in your life? Like I remember when both my children were born, exactly the moment that was. But you have like these important moments in your life where like everything crystallizes and it's like a moment you can remember. You guys have that? Okay, I'm gonna tell you a story about when I was sitting right here in 2012. I was 36 years old. I was a PhD student. I'm gonna zoom in here in the UK, just northwest of London, in a little town called Oxford. And I had a daily ritual. I was 35 years old as a first year PhD student. First of all, don't recommend that, 0 out of 5 stars. But the good news was I had enough money because I had a job that I could buy like a real sandwich every day. And so I went to this place called Taylor's, which sold these great little sandwiches. And I'm gonna tell you a couple pieces of information about this slide here. Do I have a pointer? Yeah, I do. So here is Taylor's where I went. And right across the street from that, they got so busy that they opened another store. So guess what we called that? Taylor's Expansion. Oh, Math Institute. And when I was there, the Math Institute was in this crappy, terrible building here. The Math Bio Institute was here. And the real Math Institute was here and here. But now this entire site has been raised and there's now a beautiful new Andrew Wiles Math Institute. It's freaking gorgeous. The inside, it's like an Escher painting on the inside. It's great. But I had to suffer through being in this terrible one. Anyways, I went to get a sandwich. And what I would do every day is I'd print a paper that was kind of outside my field a little. And I'd go get a sandwich and I'd read it. So I went and got a sandwich and I printed a paper. And I went and I sat right here. This little war memorial. Literally right here. My life changed. Because of this paper. Honestly. Not joking. Changed the whole way I think about the world And this is a little short piece that was written by some people in this room that looked at, does everyone know this paper? Can I just skip what it's about? That it implied this, that a parameterized fitness landscape could allow us to reproduce and predict the way evolution proceeds in a bacterial population. Please. Yeah. So what Dan and his colleagues did was took E. coli with no mutations, and then they used site-directed immunogenesis to put in 5 different individual resistance mutations to beta-lactam antibiotics, which are like a class of antibiotics that kill E. coli really well. And then that's 2 to the 5th things. It forms a complete tesseract of dimension 5. And what they did was they measured the MIC to a specific drug based on what it was. And then what they did was they asked the question, how many ways can you go uphill from the bottom to the top? So naively, it's 120 because that's how many paths there are from the bottom of a 5 cube to the top of a 5 cube. If it's Mount Fuji-like, so up, all up. But they found it's not. And in fact, they found that Darwinian evolution can follow only very few mutational paths to fitter proteins. But what that led me to do was really start thinking about, does anyone remember the game Plinko on, what was that game show on TV? Hold on, no one in this room is in my age category. Is anyone in their mid-40s? No, okay. Well, it was The Price is Right. It was basically a Galton-Watson board where you dropped a ball from the top and it plinkoed through to the bottom, and then wherever it landed was what you won. Usually it was like, oatmeal, but every once in a while it was a car, like all the way over here. But what led me to believe was that if I know how some things percolate, let me use that word that's not appropriate necessarily, but how things percolate from treatable to not, can I use knowledge of other sieves, if you allow me to call this that, to start sequencing things in order. And so let's talk about a fitness landscape. This is an idea that I stole from the internet. The idea is that we have some mapping from a genotype on the XY to fitness. And I think what Dan and colleagues showed was that it's not all uphill. More mutations, even if they're good ones, aren't always better because epistasis is a thing, it turns out. We've known this. But I'm going to skip this slide. Let's just think about this. So let's say I have a population of things, bugs, say, that are yellow. They have some genotype. They exist in this XY space somewhere. And then there's a mapping from that genotype to fitness. And so what Darwin says is it goes up, right? He was right, goes up. But what happens if it hits a branch point? What does it do? What goes up? Which way? I don't know. We've seen a couple different ways it can go on that last slide. What matters is it's become more fit. Every time you do that experiment, it's going to become more fit, but it's either going to be purple or green. What if I have another drug that looks like a different set of hills and valleys, and I give that second drug after the first one? So you're in a bad position, Isabel, because I remember your name, and Sarah, and Lissa. Yeah. Dang it! 2 out of 3 is not bad. I'm going to ask the 3 of you questions. So Sarah, if I gave this drug next, would that be a good idea or a bad idea? Why? If I went green here to this fitness peak, where would I be on this one? If I went uphill to this green star and then I gave this next drug, then I was like green star, would my fitness be high? So it wouldn't do anything. What if I gave purple? Well, still high. So no matter what, this is a bad second drug. Isabel, how about this one?
I guess that would be better.
This is like a perfect second drug because no matter what resistance mechanism you chose the first time, you're beating up. Now, how about this one?
Like which drug?
With this, what would happen if I gave this one after that one?
Gave the one at the very top after.
No, this drug, this landscape. If I applied this sieve after I applied this sieve, this is the hard one. I don't know, because you don't know where it went necessarily. So it could be a bad drug to give because it's the same peak, or it could be a great drug to give because it's lower, right? So we ended up in a situation where, what if I had two drugs or a family of drugs, could I think about how to sequence them through the lens of this theory? So what if I could parameterize this map or these maps? Could I then use them in order to take care of drugs or bugs as they evolve? Okay, so I'm going to try to answer a couple quick key questions in the next 10 minutes, and we'll talk about it. So where are things going to go? Are they going to go green triangle or purple pentagon? How fast will they go? And can we control it? So this is a first paper I wrote. Literally, I walked from that sandwich in Dan's paper to find my friend also called Dan. And we wrote this paper immediately after, thanks to what you did. And this is the paper that got me an air dash number of 3, which I'm pretty happy about because this guy is a 2. So good stuff. But what we asked here was, can we steer evolution with sequential therapies? If I have different sieves, different maps of these things, can I put them in a certain order and guarantee, or at least probabilistically bias my outcome for good or for bad? So what we did was we turned each of these landscapes. This is easy to think about, right? Hills and valleys. The problem is the genome's probably more like a bit string, as we talked about in the paper earlier. So can I write down a fitness function that takes an arbitrary length bit string from its identity to a real numbered fitness? So that's the same thing. So I could take a position here and turn it into a fitness here. And these aren't new ideas. You don't even need to know about genes to know about this. This Sewell Wright was in 1932. He came up with the idea of allelomorphs. To the people in the younger than me generation, you guys have all seen Marvel, right? Come on, you know about Tesseracts. Well, these are them. This is why. Who is it? Loki has one? I don't know. I can't pay attention. But we can now map from these long strings to fitness. So we have a perfect ability to map each of those corners to a fitness. So I can take 111, like Dan genetically engineered, measure its fitness, and write that down over here. And so fitness 0.5. And look, over here is 0.7. Those are the two highest. And if you look at the arrows, this fitness peak, this map right here is the same as that picture I drew earlier. 2 peaks that are separated, here or here. 2 possible states. You're only going to end up at one of those two. They're only 2 absorbing states. But we can now play with different versions of this. And the beautiful thing is, so we're modeling this as a random walk on this directed graph. We're going to write down as a Markov chain. Not important details, but it was important is that folks derived a whole series of these landscapes, probably from your strains, is my guess. This is a group, Marion Barlow's group from UC Santa Barbara, I think. Merced. Oh, where Amelia was. Anyways, they gave us 15 of these sieves, right? They went and measured the drug response in each of those strains across this.
So just for historical accuracy, the polarity went the other direction. Miriam gave me the system.
Oh, no way. Oh, really? That's actually very good to know.
She got it from Barry Hall, who was her PhD.
Awesome. Thanks for that. And this paper came out like right after or right before our plus CB one. It was really interesting. It was one of those things where like everyone's thinking the same thing. It's pretty fun. I'm going to skip this a little bit, but the idea is that through matrix multiplication, we can really think about how these populations emerge. So here we are with 2 peaks, and we get there through just repetitive multiplication of matrices. So what we've done is we've embedded the entire evolutionary process in a transition matrix, which is powerful because it allows us to use a lot of the tools we have for other kinds of mathematical operations to think about this process. A cute result that comes from that is that matrices don't commute, neither does evolution. If you have a species and it experiences one environment and then another, it's going to be different than a species, that same species of environment experiences environments in different directions. It's sort of obvious, but I think mathematically it makes some sense. And in bugs, it makes sense too. Because what we do as doctors is we just look up on UpToDate and pick up whatever the next drug is without a lot of rationale. Oh, you failed this first one? Let me give you the second one. Okay, so in theory, you can now steer a population with different sieves. What kind of data shows this might be useful? So I'm going to show you a really fast experiment. So this is a mentor of mine named Robert Bonomo. He's an ID doc at the Department of Veteran Affairs in Cleveland. And we did the following experiment. We took actually 60, but I'm just showing you 12 versions of the same experiment, which I'm going to call evolutionary replicates. So I took bugs, E. coli in this case. I put them on plates. So this is called a gradient plate. So you literally you just drip a whole bunch of drug in the middle of a agar plate and then put it on a record player so it spins and the drug goes to the outside because of the whole physics thing. And then at the edges, you have very little drug, and in the middle, you have a lot. And you plate individual colonies, they grow toward the middle, and you pick the thing farthest along the line, it's the most fit, it's gotten the farthest, you measure its MIC. If you do that over the course of a week or two, guess what? Everything gets more fit. Darwin was right. But it does it very, very differently because it's traversing this hidden landscape, right? So we did this 60 times for this paper. We get all these different phenomena, different trajectories. And what's interesting, not only are the trajectories different, so this is just the same picture. These are the same 10 replicates going through time over 10 days, or 12 replicates over 10 days. And we asked the question, what if we sequence them at the end? And what if we test them against other drugs at the end? So each of these replicates is now resistant to that first drug, but it's wildly different in its sensitivity compared to other drugs. So they've explored this landscape. They've all gone up. But just that picture I showed you at the beginning with the green triangle, whatever I showed you, green and purple, this second family of drugs is all different, right? So it's exploring these peaks in all these different directions. And I think that this is a strong evidence for at least the thought construct of divergence here, right? It's not saying it's perfect or we've nailed it. 2 quick slides about cancer. This also happens there. So let's look at a scenario. This is literally evolving resistance to the same class of drugs. This is single cell sequencing on a TSNE plot. It's old, don't worry too much. The idea is that we have divergent evolutionary solutions. So same idea. And we also have divergent collateral responses in this paper. So I think this, again, is evidence here. There's also big changes in radiation response, which for me is important because should I give radiation before or after these drugs? It matters, right? We don't even think about it. We just go forward. Further, this is measurable through time. So this is single cell sequencing through time of a single evolutionary replicate as it finds a peak. It's marching through some space. Again, it's TSNE. Don't at me or hate me about it. It's a hard to understand space, but the point is it's moving reliably through some space that we can catch. So again, I think there's a strong evidence there. Okay, so 2 peaks is way too simple, probably. But again, what's this balance between reality and complexity is important. And then the next question is sort of, we have to know what the landscape looks like in some way. And we also have to know how fast or how quickly it traverses that. Because if it takes 20 years to get there, who cares, right? The patient's already dead. If it takes 1 day, I can give you a blister pack of drug A, B, C, D, E, and off you go. But those timescales probably matter a lot. And so this is a 2-slide introduction to a paper that Dan suggested that they read of ours, looking at the simplest possible model of a stochastic population genetic situation. So thinking about what a population would look like on an energy surface. So physicists love going down instead of going up. So Darwin says go down in this model. And so you imagine if your energy surface looks like there's no selection, you know, kind of equilibrate in the middle. So all your things would kind of fall down into a well like that blue Gaussian. If you bias selection in one direction or the other, you'll end up with populations that kind of go each direction. And you can model this pretty easily with a Fokker-Planck formalism and end up with situations where you sort of have a peak, population will find it, stick around and follow it. And what's a lot of fun is we can now play with situations like dynamic fitness landscapes. So what if I give the first drug, finds its peak, and then I switch to a second drug and it finds the peak? Can I play with the timing of my administration of these in such a way that the bugs get behind or that I stay ahead in a different way? And this is all made possible by work, I think, done here by Brandon Ogbunu. Was this when he was a postdoc or what's the history? He was at Harvard. This is done by a person who was here for a while before he got here in a malarial system. And this is really important. This is the first fitness seascape that I've been able to find and parameterize. And these are actually the data we used. And the idea is that for each different, again, looking at these mutational types, their fitness changes not just with what type they are, but with what dose of drug they're getting. So it's not just the drug, it's how much. Just like I showed you on that plot of the evolutionary games, it's not just the drug, it's how much. It moves it through that evolutionary space. And Brandon showed us the same thing in this malarial system. And this matters because if you have a funky trajectory like this, or a funky seascape like this, as we call them, as patients take drug or forget to take drug, we kind of climb toward a steady state with some pills. We forget for a while, we climb back toward a steady state, we forget. What's happening to the drug concentration as they take those pills? It's traversing this space and it's going high and low and it's switching the landscape that they're experiencing. So at the beginning, you might have one landscape and favor this mutant here. But as you forget your drug, maybe you're favoring a new mutant. And you're actually asking that population to explore this space as you move forward in time. That's horrifying to me, because these are natural perturbations and excursions through dose space. Even if this patient took all their drugs, it's still having an excursion here. What if that range happens to be right in the middle of where these evolutionary trade-offs occur? We're not even thinking about it. All of a sudden, we're stirring the pot of evolution, right? And I think that that's just something that's worth thinking about. And here's just a single snapshot of an idea, and I'm going to play a video a couple times in a row. So what you're looking at here is, so I just showed you a second ago that as the dose changes, the landscape changes, right? And this is a simplified 1D version of that to make it a little easier. So this blue trace is dose slowly changing from low to something. And this red trace is dose changing from the same low to the same something, but through a large excursion. And this was specifically derived in order to speed up the flow from where we start to where we want to go. And what you can see on the bottom is that as the dose changes, this red is snapping to, see underneath it, there's green. That green is the predicted final steady state. And the red follows it, whereas the blue with this gradual change takes forever to get there. There's a lot of beautiful literature out there, Ben Kerr and some others looking at slow versus fast adaptation. And in these slow adaptation situations are very different. So the evolution giving it more and less time. So if we can snap something from where we want to start to where we want to go, we have a better chance of control. And so we go into a lot more detail in this paper thinking about how control could work on these hypersurfaces. And that's Emily Dolson and Mike Konchesky that would work. Emily's a professor at Michigan State in computer science, if you're interested in working with an amazing woman who does evolutionary algorithms. And Mike's a physicist at where I work, Case Western. Okay, that's the end of the middle, and there's 2 minutes left. So I just showed you all the stuff about games and how I could push stuff around and how amazing it was. I showed you all the stuff about landscapes and how you could push stuff around and how amazing it was. But I promised you at the beginning that I shove them together, and I didn't, because there's different timescales. So where's the landscape in the game, and where's the game in the landscape, and how can we get there? So let's look at really quick the equations that we use to find our game. So this is the simple version where I've already normalized, and I have just my 4 parameters. But there's no landscape there, right? It's just these interaction terms. Where the heck is this information I just showed you that was so important? Well, let's separate out those terms a little bit. So if you want to, you can start normalizing that payoff matrix and bring it down to just an intrinsic growth rate of the parental, A, just an intrinsic growth rate of the mutant, D, and now modify your diagonal terms to be functions with positive offsets, where this is just the interaction. So what I showed you how to measure was this, 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 and this. What Dan and others have showed us how to measure is this, and by extension, this and this. So what we're suggesting is we measure both. We can separate them out and actually come up with a single formalism that allows us to understand how both the interactions and the games work together. So we understand intrinsic fitness, and we understand the off-diagonal terms, which also bring in this ecological dynamic. So now we can go back to our original stuff, measure the things I showed you, and now write down a sort of master governing equation for the eco-evolutionary dynamics, which I'm hoping are our next step. And this is work that's not yet out, but it's written by an unbelievable postdoc I have called Rowan, and it's on bioRxiv now. You can read it. It should be out soon. You end up with similar results, but it's shifted in a super meaningful way. And we're really excited about this. The last piece of the puzzle is so I've shown you how to measure games in cancer. I've shown you landscapes measured in bugs. And for the last 4 years, this guy, Dag, who is now an assistant professor in Oslo, who came to me from Oslo after he finished his PhD to postdoc, spent 4 years in my lab building the first hypercube in cancer. And so we now finally have a 4-cube with the 4 most important mutations for EGFR resistance, which are over-amplification of EGFR. So this is an ancestor PC9 non-small cell lung cancer, an amplification of EGFR, an addition of PIK3CA, an addition of KRAS, an addition of BRAF, and everything in between. It exhibits all the same phenomena we expect from Dan's work and others, where the growth rate declines with more mutations. The drugless growth rate declines. So we have this cost of resistance. We have an increase in a decrease in drug response, or sorry, an increase in the IC50, so increased resistance. And we have our first hypercube. So Dan said, measuring this implies the protein of life might be reproducible, predictable. Here's our first hypercube in cancer. And I wonder, might also it be controllable? And I think we can only get there if we take the lessons we've learned from the fitness landscape work we've seen in bacteria, hopefully the game theory work we've been doing in cancer, and bringing those all together into a single formalism. And I'm hopeful that we can start showing perturbation experiments where we can control what's happening on these. We have the mathematical tools. As of a few weeks ago, we have the biological tools, and I'm super excited for what the next steps might be. And that's the end. And I'm only 4 minutes late.
One of the earlier slides, in the last part, you mentioned that you used a random walk.
Yeah.
Why did you use a random walk as opposed to some sort of weighted probabilistic model?
So depending on how you look at it, I'm just going to find this one here, right? This one. So it actually ends up that the random walk is a weighted probabilistic model. So it's specifically that. So the randomness comes from the probabilities transitioning from I to J, and there's this little guy up here where, depending on what you put that R term to, it's either purely greedy, totally random, or biased. And so the biased random walk is what we used. The results actually hold for all three. The qualitative results hold for all three, but the quantitative aspects are different. But we played with a biased uphill walk, which is, I think, what we learned from Dan. This is sort of transition rates based on that biased probability, right? Same formula, yeah.
Except without the R.
Was that?
Without the R,
You just left it off. Well, I said it's a 1. Yeah, so mine's more complicated. It has an R in it. The R was 1. I didn't invent anything. I just stole it from people. Sorry, I stood on the shoulders of giants. Any other questions?
So is this, like, would this result in balancing selection? Environment, always situation. With a drug treatment program, in practical terms for patients. The outcome would never be to cure the cancer, it would be to control it back.
Yeah, that's right.
That means that they would be on drugs for the rest of their lives, but at least their life would be wrong.
That's right.
Maintain polymers. Acceptable existence of cells,
That's right. The more hopeful version would be you keep it at such a low level for long enough that the immune system figures it out. Right. And so I didn't mention immune oncology anywhere here, but, thinking about if I could get this down below X% for long enough, we have all these checkpoint inhibitors and other drugs which prime the immune system. Maybe with enough time, area under the curve of surveillance, we could get there. But yes, the premise that I'm putting forward without any of that is chronic disease.
What's the actual tumor burden?
No. So in the first part of the talk where it was all the gamesy stuff, that's all actually not even lack of Volterra, so that's all normalized to 1. So no. But I think that the second formalism allows us, and we can always switch it to a model that allows for proper growth. But at the time, no. So we have a grant right now that's trying to look at
If you want to balance, but also.
that's right. Oh, I didn't include that slide here, but we have a grant we're working on right now to try to put this into a non-replicator dynamic, so more like lack of Volterra, and then also into an animal model. So then we can actually have proper growth rather than right now it's just a dish, right? So we're stuck between 0 and full.
It's one thing to start from a single background and apply formulations, but
Yeah. Yeah, I would say this is exactly what we were talking about at breakfast, right? This is the art of the modeling. So that, and I go back to my analogy about the wolves and the moose, right? There is some sweet spot of simple that still works. But I'm really nervous that you know there's more than 4 mutations in lung cancer, right? Turns out, and probably many that are coexisting, right? So this is also still strong selection mutation, random walk through this directed graph. What we're thinking about right now, so the experiment we're doing now, I can draw on the wall, right? So the experiment we're doing now, what I'm excited about is, so let's just think about the simplest version. Is so that this is now the same scenario, right? This is the same tesseract. What we're asking is, so what can happen here? Well, you can go that way or any other way. That's the problem, right? So in E. coli, how many genes are in E. coli? 7,000? A lot. No, how long is the genome of E. coli? I think it's 70K genes, or base pairs. So was that
4 megabits.
10 to the 6th? So
4 million base pairs, you get 1,000.
4,000K. It's a lot. Okay, so you can go this way, or 3,999,000 other ways. Yeah, 1,000 other ways. So what we're doing right now, so have you guys heard of this whole literature on DFEs, or distributions of fitness effects? Does that ring any bells? So it's something called a DFE. And what they do is so Vaughn Cooper's really good at this, a bunch of other folks are. They say, what does the fundamental distribution look like of fitness versus mutations? And this is a KDE that shows density. This is a probability density. This is a probability density of mutations and how frequent or what fitness benefit they have. So what we're trying to do experimentally right now is, okay, hey, and we're using your cell lines for these that we got from you. One of them's contaminated, not one of these 4. I might need to get another copy. What we're doing is putting these guys in a dish under no selection and asking what this looks like. So we're doing, so we just did 100 drift experiments, right? Did 100 replicates of no drug here. And then I plotted all the mutations that occurred, and we haven't measured their fitnesses yet, but we have some distribution. Well, we don't yet because we haven't measured this part yet. But the idea is we're going to get some fundamental distribution, or we're going to hypothesize that the distribution we measure is fundamental. And we're going to do the same thing over here. I don't know, maybe it's the same thing over here. Maybe it's different. Same thing over here. So we're going to have 4 DFEs. And then what we're going to try to do is say, okay, my fundamental fitness, it used to be, Dan said 4 or whatever. So I can just call that some function of, you call this some, the fitness at 0, 0 is some, the fitness at 0, 0 is some function of its position, so its genotype, plus some other function of the DFE times the number of mutations, say. And so what we're trying to do is parameterize. So we already, this we know, because Dan measured it. And now we're going to go measure this and then ask the question, you know, can we now put a stochastic process on the whole thing? So now what you'd have at the end of that is, as each corner, you'd now have to loosen your assumption about strong selection mutation, but now each corner would end up with a distribution of stuff. And that distribution would be, so it would no longer be how many things are at 0, 0. It would be how many things at 0, 0 and what is their distribution of that fitness. So that's how we're trying to get around some of it, because down the other road, madness lies, right? Because it turns out I just showed you Doug, who's like the best postdoc on planet Earth. And it took him 4 years to make that thing. It was just 3 years a minute ago. I'm just hyperbolic. Took him 3 years to make that thing, and it's a 4 cube, right? So if I'm like, hey, Doug, go make a 5 cube. Guess how combinatorics work? Not good for Doug. So I think what we're hoping is to kind of find a parsimonious way to parameterize an infinite dimensional version of this. And then the idea would be that the leading order is the N mutations that I think are most important. But that's a hard question. The other thing I'd say to someone skeptical is like, where's your imagination, man? What else? Thank you.
It's possible that that, like, is there some sort of, like, in the diagram you showed, like, signal slows down and expands depending on like, like I forgot. What is that?
In the first part of the second
Yeah.
part. Oh, this whole like ice cream cone?
Yeah.
Okay.
So I guess like, with the combinations of treatment plans, is there like some sort of threshold of solid?
Oh, yeah. Great question. So, I had a postdoc named Nara Yoon, who's now associate professor at Delphi University, who wrote a paper that I'll share, where she coined the term shaping heterogeneity. And the idea was, right here at point of max diversity. Okay, so where are my wolves and my moose here? Okay, so here, wolves, moose, right? Now I've got wolves, moose, and toucans. And now I've got wolves, they don't call me. But what do I have here? Like too much, right? So one skeptic might say, hey, your 2 population control model is going to fall down here because there's just too much stuff. There's too many things chasing the wolves or whatever. So one idea would be, can I force this into some sort of functional bottleneck to reduce that heterogeneity from here to here, such that I have a control list. So we call that shaping the heterogeneity. We've made it. But yeah, I think that's an idea, is to sort of control the complexities. Yeah, it would be sort of doing as well as you can to begin with, and then seeing what you have left. The other way to look at that is like, this is just in cartoon, obviously, but maybe it is enough to know the 2 leading terms and control them until they no longer matter, and then you have more leading terms to switch.
Based on the state of the research right now with this research, what
Good meeting.
can physicians do right now to go and implement this to make their treatments more effective?
Yeah, great question. I think the answer is, so I'm torn by the way I want to answer that. So there are clinical trials being run now of this game theory stuff, like I showed here. There's like 3 or 4 active trials now at Moffitt Cancer Center, different cancers, kind of guessing this and doing it. That makes me nervous because, so you're way too young to remember, but when we first started doing gene therapy, the first one was a success, and everyone raced to do more, and they started killing people with bad vectors and bad protocols. And that put like a 30-year moratorium on gene therapy. I am nervous that this first trial, which has fanned the flames in the field of people now writing the crap out of these trials, I think it's done. I personally am nervous because I feel like we have to now go back. I think we should use this to get lots of grants and excite people and go back and do the bench science so we can understand it, because it's pretty easy to write down a different version of what they wrote down and get a really different answer. So my feeling is we probably shouldn't be changing so much right now outside of at least trials. So standard of care, I wouldn't change any of based on any of this. But there is sort of a new subfield called evolutionary therapy that's trying to make people think about it.
Where do you think, I guess just off the record, where do you think this is gonna go, and where do you think that it's, just based on your hunch, physicians will be able to?
So we've written a bunch of grants and proposed trials of adaptive therapy. And I do think in lung cancer, at least, we have a handful of papers now that show that it might be feasible. The question is, how do you monitor the populations in real time to see if it's frickin' working? So again, one of our grants that we have active now is trying to make an animal model of this. And the idea is going to be to use a blood-borne metric of levels In an animal model, that's easy because you can take the red and green cells and make them glow with different luciferins. That's harder in a patient, but ctDNA does work. And there's a really gorgeous paper from this group that worked to use so liquid biopsies through time, so blood-based biopsies. And they this is a really cool group if you don't know these guys. Andreas in Milan, and amazing, and Ben Werner, who did all the real work for the paper here, is at UCL as a mathematician. What they did was they fit the simplest darn model you can imagine, exponential growth, to, well, I'll let you read it, but they fit an exponential growth model and use ctDNA there, a banger, to ctDNA, and they actually predicted responses before they were seen on scans or anything. So this gives me hope that this kind of longitudinal liquid-based biopsy can give us enough insight into the underlying populations that we can start playing with them. So I think it's not infeasible by the time you're a physician that this is real. How old are you?
I'm a freshman.
In high school or medical school or college? Okay, yeah, maybe. Yeah, there's a chance. If you said med school, I was going to say no. But cool. Thanks, everybody. Oh, you got one more? Yes. Oh, we're certainly neglecting all the things that I didn't write down. I mean, there's a million things that, you know, that it's hard to include everything for sure. Well, it's possible. Where'd my talk go? You mean this sort of final hopeful equation at the end? I think that, so Rowan, have a read of this paper, check it out. What I think is cool about it is that it is eco-evo, the timescales kind of overlap. Is there stuff missing from the model? Yeah. All the off-landscape mutations, all the other populations missing entirely. I didn't use the word mechanism once, which is like antithetical to a biology talk, but I don't really care what the mechanism of it is if I have the phenomenological outcome of that mechanism. I could have told you that it was mediated by hepatocyte growth factor secreted by the whatever, and it doesn't matter. But I do think, you know, another thing, I'll just say one more thing and then we'll all go somewhere. The question nobody asked, which is that in all of our antibiotic experiments that I showed you, like these ones, evolution did different things every darn time. Right, but no one said, what if you did this equation or this evolution experiment more than once? What I told you was I evolved resistance and then I competed the resistant versus sensitive and I got a game. Can I trust that the next time I evolve resistance, I get the same game? I don't think so. We're doing this as we speak. So we actually have a PhD student named Mina, who was on one of those slides, whose PhD is all about, young lady. She's evolved 10 replicates to osimertinib, and she's playing all those games back against the parental. Question, are they the same? I don't think they're going to be. Then the next question is, can I map from their transcriptome to the game? So building a map from transcriptome to game space would be great, because then I could biopsy you and maybe pull a game map out, but far away. Thanks.
Thank you. Thank you.
Sorry for going over. Or better yet, thank you for letting me go over. Thanks, Zoom audience.
Thank you.